All right, folks, we're sort of in the final stretch. I want to welcome you all to the Open BME seminar series. This is the second to last seminar series for this semester. It's gone so well. I hope we can continue this in the future, but we've got today. So if this is your first seminar, if you've attended multiple, you've probably heard of Open BME. This is a consortium of 7 of the top BME programs in the country. You see the logos here. So we got together thinking that as a cohesive unit, as a consortium, we could attract some of the top speakers in the world to come to our seminars series and take advantage of the Zoom environment we have now to allow that speaker to interact with faculty and students from all these different schools at the same time. And the benefit is you get to see it through live through this Zoom teleconference. It's also being streamed to YouTube and can be seen later if you happen to be seeing this asynchronously. So just a quick orientation, there's a Q&A feature. So you won't be able to chat directly with the speaker. That the chat is disabled, but there's a Q&A feature where you can ask questions. I encourage you to ask questions. The Q&A is along the bottom row. Just feel free to type in any questions you have as they come up. And the neat thing is, even if you don't have a question, you should check out the Q&A because you can upvote questions you like. So if someone's asking something and you say, hey, I'm interested in that, you can click on that question, the thumbs up, and raise that one in priority. And at the end, the speaker will go through the questions by priority ranking. I will probably 10 minutes, 5 minutes before the end of the talk, I'll send out through chat a reminder to remind folks to do Q&A. Sometimes the chats are so captivating that people forget to do that, but just try to remember to do that. And with that, I'm going to introduce Professor Lonnie Shea, the chair from Department of Biomedical Engineering at University of Michigan.
Well, fantastic. Well, thank you, Ars. Really my great pleasure when we were thinking about organizing this session today. We wanted to sort of bring someone in at the interface of tissue engineering and synthetic biology. I think the idea of tissue engineering is obviously it's a really well-established component of BME, but hasn't really had those sort of breakthroughs in terms of its developments industrially or in terms of helping patients as well. And I think synthetic biology represents sort of some new tools that could come to bear to really open up some really exciting new opportunities for tissue engineering and cellular engineering as well. And so with that, I'm really fortunate to be able to welcome Professor Tim Downing here to speak to us on this very general topic, who has really been making the focus of his lab is really as the intersection, as you can see in this title slide. So Tim is currently an assistant professor of biomedical engineering at UC Irvine, also with a curacy appointment in essentially molecular microbiology and immunology. And at the same time, he had some very humble beginnings at Northwestern University, actually in my lab. So it's maybe my fault that he is had some small component to this, but at the same time, he went on and did some really outstanding work at UC Berkeley for his PhD, working with Song Lee, and then progress on to do postdoctoral work at the Broad Institute, really look at epigenetic programming and epigenetics in terms of cellular reprogramming. And so some really exciting work. And then obviously, since his time at UC Irvine, has really worked on this idea of synthetic genome engineering. So with that, Tim has received a number of awards as well, too, in terms of a Merck Fellow. He's most recently received the DP2 award for his work, and also is sort of a rising star by the Cellular Molecular Bioengineering group within BMES. And so it's really an honor to have you here, Tim, and I will turn the floor over to you to sort of share your exciting work. Thanks for joining us.
Wonderful. Thank you so much, Lonnie. It's a pleasure to be here and really to share my work with such an incredibly diverse group of people across 7 campuses. I'm going to share my screen here. Let's see. Okay, you can all see this. Great. Yeah, so as Lonnie mentioned, I'm going to share really a story and hopefully a second story about some of the things that have been going on in my lab around this idea of ultimately thinking about synthetic genome regulation for cell and tissue engineering. So one of the things that my lab has always really been interested in is understanding this question of how do single genomes beautifully give rise to all of the cellular heterogeneity that exists within tissues. And so here's a developing embryo and looking at this intercell mass. All these cells have the same genetic makeup, all throughout our bodies, but they're able to give rise to all of these different tissues. And what the field has really come to appreciate is that a key way in which cells can do this is through epigenetic regulation. I'm really referring to these chemical modifications that happen on top of DNA or proteins, like histone modifications and the tails of those modifications, as well as things like DNA methylation, which will be the focus of my talk. These things play a big part in the regulation of actual gene expression, which ultimately gives cells protein expression and function. And so when I got into this field, I think a lot of what was focused in on chromatin dynamics and understanding epigenetic regulation was done at the level of cell types. So understanding how epigenetic changes take place as cells transition from one cell type to another cell type. But the other part of this that really fascinated me was that every time a cell replicates, in order to remember which genes are sort of should be on and left on versus remaining off, the cell also has to reestablish and replicate this epigenetic landscape, if you will. And at the same time, we know that replication is this sort of molecular foothold that cells can use to move through different cellular states. Things like stem cells, asymmetric division. We know replication is a really important part. But also there have been observations where populations of stem cells will behave differently to a differentiation sort of cue from the environment, depending on which stage in the cell cycle those cells are in. So here's sort of a schematic from Stephen Dalton's review here, showing that cells in G1 will upregulate developmental genes after some time of induction earlier than cells that are, for example, in G2 or S phase. At the same time, there's this sort of duality of replication where we know it's important for stem cells' ability to make sort of fate decisions. At the same time, replication also seems to leave our cells susceptible to aging and really drive mechanisms of aging. And sort of one of the well-known examples of how replication sort of facilitates the aging process is through telomere shortening. Obviously, with linear chromosomes, that sort of comes with the cost of this in-replication problem where telomere shortening happens at the ends of chromosomes if telomerase is not working to extend this. And so my lab really is thinking about how replication can serve these sort of dual functions and the utility in that. But in particular, we're very interested in epigenetics from this lens. We theorize that temporal delays in epigenome replication actually serve dual functions in mammalian cells. And I'll try to convince you of that to some degree in this talk. But we first think that these temporal delays actually it introduces temporal regulatory variation in stem cell populations, which I think many would argue is a critical feature of multicellular life. But it also confers susceptibility to a progressive drift in the epigenetic landscape throughout the lifespan, this sort of epigenetic drift observation, ultimately facilitating the aging process. And so why this is sort of important and relevant is some theories on aging actually argue that aging emerged early in our evolution as a means of controlling population size. This idea of how to achieve a sort of ecological steady state with an organism growing in an environment that's also consuming resources. And so as resources come in and they get consumed, population grows without a death mechanism of that organism, your resources would be out competed by that growing population, and then the population would follow behind and crash out. And so I love this quote from Teschendorf, in our ancestral species, natural group selection could have favored genetic and epigenetic mechanisms that actually promote aging. So my lab is interested in, again, synthetic genome regulation for cell and tissue engineering. More broadly, we're very interested in how chemical and biophysical cues from the extracellular environment actually influence cells in the context of many different sort of biological and biomedically relevant applications, things like cell reprogramming, taking fibroblasts back to iPSCs, tumor growth, and particularly cancer stemness is one area that we're very interested in, as well as aging. And we're also interested in how we can use this information to design better biomaterials for things like wound healing and implant biology. But today I'm going to talk about particularly the mechanisms that we're interested in within this paradigm are chromatin dynamics throughout the cell cycle, and really thinking about epigenome reassembly as a sort of window for cell fate regulation and ultimately cell fate control. So looking back to this sort of picture of the epigenetic landscape, again, what has to be mitotically heritable, things like DNA methylation, histone modifications, composition and position, and nuclear architecture. All of these things are starting to emerge as critical epigenetic and chromatin features that really drive cell fate. And so DNA methylation refers to actually a cytosine on the cytosine molecule or base within DNA. It refers to this methyl group that gets added to the 5' position of the cytosine. And just to give you a scale of how many sites within an human embryonic stem cell that actually sort of exist and actually have to get reestablished every time a cell replicates, there's 56 million CpG sites in the diploid genome, and 45 million of them have some level of methylation. So there's just an incredible amount of enzymatic activity and work that has to be achieved for these cells to actually maintain this epigenetic modification across replication. And so here I'm showing sort of this relatively gold standard in the field now with analyzing methylation data using something called whole genome bisulfite sequencing. The way that it really works is we do a bisulfite conversion on DNA, and that changes unmethylated cytosines to effectively Ts when we send them to the sequencer. And with that base pair change, we can know the methylation status of every cytosine that we sequence. And then if we capture a cytosine in a given location in the genome at a certain depth, we can calculate a methylation ratio. So on the far right here, you're seeing a scenario where you have 5 cells that exist that you may have sequenced DNA from, and 6 out of the 10 CpG sites actually have some methylation. And so you get a methylation ratio of 0.6 or 60%. And you can get other sort of intermediate methylation levels. And so when you look at this data and what it actually looks like in unsynchronized hESCs, you can appreciate that even though this is a homogeneous group of cells, you'll have some CpGs with a high frequency at methylation ratios of 0 and also 1, but you also have quite a few that exist in this intermediate space. And the field, because methylation is binary in its nature on a single cell level, I think the field has really thought of this modification in a very binary sense. And part of the reason for that is when you look at these on a genome browser track, so here now we're showing a region of the genome for this UBE2J2 gene, and methylation ratios on the y-axis, where every dot is a CpG along this genomic loci. You can see it sort of has this very binary nature, but there is indeed a degree of heterogeneity that exists within these methylated cytosine methylation ratios. And so part of when I started my work in my postdoc, we wanted to ask this question of what could be driving this heterogeneity, and could it have some biological function or meaning to actual stem cell fate decision makings. And one thing that we thought about was replication as being a source of some of this heterogeneity. And so just to give you a little flavor of how DNA methylation is really propagated across cell divisions and DNA replication, it exists, and we see it most frequently again in this CpG dinucleotide context. And the fascinating thing about this is that it's a palindrome-like structure. The CpG is the same as it is in the forward direction as it is in reverse. And so there's this opportunity for cytosines on both forward and reverse strands to be methylated. And every time a cell or DNA is replicated, actually DNA polymerase won't use this methylated cytosine as a substrate. And so you have this period of time where DNA is actually unmethylated. And there's an enzyme, DNA methyltransferase 1, that has its catalytic activity almost exclusively reserved for adding methylation to hemimethylated DNA strands, ultimately leading to a fully methylated strand of DNA. So in a sense, you have this closed loop system where DNMT1 is always bringing this double-stranded, fully methylated strand back across replication to a fully methylated state. So during my postdoc, we developed this method called Repli bisulfite sequencing to measure methylation in newly replicated DNA over time. And so the way that it works, very briefly, is that we take embryonic stem cells growing asynchronously in a dish. We post-label them with a thymidine analog BRDU for 1 hour. So actually a lot of DNA can get labeled in this 1 hour span. And long story short, we can fish out these newly replicated strands by using a BRDU antibody through an immunoprecipitation effectively. And then we can do bisulfite sequencing to measure methylation in that. And the really striking thing that we observe from this analysis is that the green line here is actually showing what we traditionally will measure in whole genome bisulfite data in cells that haven't been synchronized. And you can see that there's some variation. But when we look at the newly replicated strands from our BRDU-IP, you can see that even after this 1 hour pulse, and again, polymerase is moving quite fast, sub-second sort of every base is a sub-second sort of kinetics, we still see this global hypomethylation and DNA methylation in that newly replicated strand shown in purple. And not only that, when we arrest cells in G2M phase using the cortisol, we can see that there's a hypermethylation globally that happens as well, really suggesting that what we've been measuring before is a blend of DNA that is newly replicated as well as DNA that's been more matured. We can do a pulse chase experiment actually and watch these changes over time. This is showing an example where we pulse label this with BRDU, and then we can follow this over time through a chase period. And what you can appreciate is that even after 1 hour after our pulse, we're still hypomethylated. 2 hours, we still have some level of hypomethylation. And not until around 4 hours, we start to hit those normal levels. But even beyond 4 hours, there seems to be some increase in methylation that we can observe that's happening in these replicated strands. And so it really paints this picture of adding really new known sources of methylation heterogeneity, that it's not only just stable heterogeneity that exists stably across different cell types, but actually if you capture different cells in different places in time after a region was replicated, and certain regions are very slow to be remethylated, and some may be even faster, and some may be very fast in their remethylation, you can actually recreate these methylation profiles that you commonly observe within whole genome bisulfite sequencing data. So really this temporal delay in replication seems to be a key driver in driving methylation heterogeneity in embryonic stem cells. So in my lab, we've been really trying to answer this question of, does this have some functional consequence in the things that we think are important for stem cells? So things like gene expression and gene regulation. And one of the ways that we have been looking at this and approaching it is asking, well, let's look at the difference in methylation ratio from when cells are at this steady state 16-hour state, and that's shown here in this dark purple line, versus that 0-hour nascent DNA line. And what you can appreciate is that there's some variability, even though all of these end up in the same place in steady state at that 0-hour time point, there seems to be a clear difference in sort of the kinetics in which they get to that steady state place. And so can we learn something about what might be happening in terms of the heterogeneity across the population through these sort of dynamics? And so the way that we do this, or one of the ways in which we've been doing this, is by creating these temporally dynamic tiles, as we call them, where we focus on regions that are highly divergent from 16-hour to 0 hours after the pulse chase experiment. And so regions that are very highly divergent between 16 hours and 0 hours are represented in our highest percentile bins of dynamics. So we call those Repli-DMRs, representing 90 to 100 percentile bins. And then we can have some that are obviously in lower, less dynamic and representing in sort of the 0 to 10 and 10 to 20 percentile bins. So what does this lag in post-replication methylation actually mean? Well, it means that methylation is there in some cells within that population, but not in other cells, okay? And the greater the difference in steady state and post-replication methylation is, the longer those differential states in methylation exist. And so you effectively will have this persistent temporal heterogeneity in methylation across a cell population. And so again, some cells can be absent of methylation while other cells actually have methylation. And you might even have another location in the genome where methylation is absent in those cells while being present in other cells. And so this is sort of like a state-like pattern that could emerge within this epigenetic landscape, again, driven by post-replication epigenetic activity. So this is idea of accessibility or epigenetic changes that are happening throughout the cell cycle has actually been observed in other contexts. This is one of the nice ones, and my favorite ones is sort of showing attack-seek data, looking at genome accessibility throughout in cells in different stages of the cell cycle. So we're looking at cells that are non-synchronized. So traditionally what we're looking at in the red bar, usually in a lot of contexts, you can see that there's a high level of accessibility, particularly in weak enhancers and CTCF insulator sites and other segments, as well as active promoters. But then when you look at cells that are in a mitotic phase or have a mitotic chromatin state, you can see that these accessibility points actually go away and don't exist. So one of the things that we started to do with our Repli-DMRs, again, these replication associated differentially methylated regions, as the sort of full name is, we wanted to see what they associate with in terms of chromatin features. Just looking at sort of things in hESCs like enhancers versus promoters and other genomic features, we see that there is a slight fold enrichment that we see in these Repli-DMRs in enhancer regions. And that actually is corroborated with some of the histone modifications that they also seem to be enriched with, particularly H3K4 monomethylation, which is oftentimes associated with types of enhancers inside of the cell. So these enhancers are places that can actually regulate and enhance the expression of genes nearby effectively, and plays a big role in the way genes are regulated in embryonic stem cells. The other piece that we wanted to sort of look at, and I'll touch upon a little bit here, is we wanted to see how regions that we know are dynamically regulated in DNA methylation as embryonic stem cells specify or differentiate into these germ layers, ectoderm, mesoderm, or endoderm germ layers. And we wanted to see what relationship they had with those, particularly as it might relate to this idea of a window of opportunity of this delay in methylation actually being a window where fate may actually be coaxed to change if there's a moment where methylation is not present in that location. And so looking at traditional DMRs, so differentially methylated regions, again, that have been defined by changes that occur as embryonic stem cells change from their state to an ectoderm state or mesoderm state, et cetera. Those DMRs also show some fold enrichment, and we see a particularly interesting fold enrichment of almost 7-fold in the case of ectoderm locations, that these are places where DNA becomes hypomethylated relative to embryonic stem cells during differentiation. And so we seem to be highlighting or enriching for this population by focusing on these Repli-DMRs. Furthermore, when we do go-term sort of analyses of biological processes, we also see that there's a strong association with terms associated and genes associated with development and differentiation, anatomical structure development, system development, cell differentiation, cell development. And we also see a lot of terms related to nervous system development as well, which also is corroborating with some of our enhanced enrichments that we observe in the ectoderm DMR case as well. But I want to talk a little bit more about sort of the nature of the methylation that we observe in these newly replicated strands, to really hit on this point of, is this a state-like pattern that is emerging in the epigenetic landscape, or is there more of a randomized pattern where intracellular methylation might be high, variation within the same sequencing read effectively, or is it more homogeneous within a methylation read and the divergence actually happens across cell types? And I'll just point out both of these cases will actually have very similar methylation readouts. And so the only way to do this is to actually do a single read analysis of the actual data. And again, the implications of a cell state-like pattern would really support this idea of this being either an indication of regulatory factors or transcription factors that are able to bind or unable to bind to a given location and may compete with methyltransferase enzymes, for example, as well. So the way that we started to do this is actually developing something called a transition score, where we look at how many times a transition in methylation happens on a single read. And so for every transition from unmethylated to methylated, we add 1, and we can add 1 again if it goes from methylated to unmethylated. And if there's no transition, we don't add any number. And so with that, we can take an average effectively of those transitions and come up with this transition score. At the same time, we can create synthetic data where we can apply methylation to match the same global mean methylation levels of newly replicated strands, but apply it in a very different fashion. We can apply it in a state-like pattern where transitions are low, or we can apply it in a randomized pattern where methylation is applied randomly and transitions are high. And you can appreciate the distribution of transition scores that we can get in that context of the randomized methylation pattern versus the cell state-like pattern. And here's sort of an example of what the actual data looks like, to give you a sort of hint. But to quantify it, we developed we analyzed those distributions through a Jensen-Shannon divergence calculation. And so the lower the score, the more similar the profiles are to the actual data in this case. And so what you can appreciate is actually throughout all of our temporally dynamic tile percentile bins, from 10 to 30%, and even more so in the highly dynamic tiles, we actually see that the state-like model is more similar and has less divergence compared to that randomized pattern. If we look at reads that have more CPGs or based on the number of CPGs, actually there seems to be a bit of a bias in the actually the randomized pattern being more similar to the data. But as you get to about 5 CPGs in the read, the divergence really starts to take over and really shows this state-like pattern is the dominant mode in which methylation seems to be applied within these sequencing reads. And so again, this really supports the idea that this post-replication activity occurs in a state that would favor more of a cellular state-like pattern where gene regulatory states are different between different cell types rather than, again, randomized. So I also want to talk about another way that we've been looking at this data using stochastic modeling, actually, and maximum likelihood estimations with a collaborator at UCI, Assistant Professor Elizabeth Reed. We've developed a new kinetic rate parameter, K, for each individual CPG captured during our post-chase experiments. And this metric K actually describes the rate at which each CPG that we capture with enough depth in data, actually the rate at which that CPG across the population achieves steady state methylation levels, okay? And so the slower the K, the more persistent, again, that temporal regulatory variation would be across the population. And this is a really cool sort of tool, because now we can get information on single CPG resolution rather than relying on our temporally dynamic tiles, where, like traditional DMRs, they actually rely on sort of local activity of CPGs. And so we don't get single CPG resolution in those sort of measurements. And some of the very interesting things that we see using this new parameter K is that we can find enrichments for certain regulatory factors that bind to DNA. One that seems to be really pronounced, and again, speaks, I think, to this association with enhancers, but this P300 gene, which is a histone acetyltransferase that oftentimes is associated with enhancer regions. But also we observe and we can find distributions of how Ks are distributed within different transcription factor binding locations in the genome. And we can compare this to the genomic average shown in dark gray here. But what we notice is that regulatory factors important for stem cells actually have a much slower kinetic profile, and they have a much further Jensen-Shannon divergence compared to other transcription factors that might exist within publicly available data sets of transcription factor binding sites. So here's GATA1, and it seems to match very similarly to the genomic average. But things like NANOG and OCT4 and a few other very interesting sort of hits seem to actually be very divergent and highly divergent from what other regulatory factor binding sites look like. So one of the things that we also wanted to ask about this data is, well, could there be some indication that this has a regulatory impact on gene expression? Because ultimately, we tend to think of that as being really important for controlling cell fate and behavior. And so we use single cell RNA sequencing data to look at gene expression to ask a question about, well, a hypothetical case, if we had a loci that had slow remethylation kinetics, that would mean that there was more variation across the population from cell to cell near that gene effectively. And that might be reflected in differences in the expression of that gene across those two cell types versus a location that has fast remethylation kinetics. There would be less temporal heterogeneity for that gene region. And that would look like less variation in gene expression versus the other case that has a higher level. And so using this single cell RNA sequencing data set, we clustered genes according to their variability and expression. And what you can appreciate from this. And on the y-axis, again, is this parameter K, rate of remethylation. The slowly remethylated rates, the areas with low variability in gene expression between cells, again, has faster remethylation rates around that transcriptional start site of those genes, versus regions or genes with high variability actually have lower, on average, remethylation rates around that gene. And you can see sort of this really nice sort of trend that seems to appear and emerge as you go from high to low variability in a gene's expression, really supporting this idea that this local activity could have some impact on gene regulation and expression in this post-replication space. We've been doing some single cell attack-seq and single cell RNA-seq in the same cell to really tease this apart in the same cell. And so hopefully I'll have more to share about that in the future. But the reason that we're really so excited about this is that it appears that this post-replication methylation heterogeneity could be a contribution to this world of molecular variability that's emerging at all levels of the central dogma, from protein levels to RNA, and ultimately to gene regulation, that's been recently showed in things like single cell attack-seq data. But the source of this variation is sort of really poorly understood. But what is starting to become appreciated is that this molecular variation could be useful for stem cells to accomplish tasks that are really important for their function, particularly things like probabilistic differentiation. So in the context of having a stem cell being challenged or exposed to a differentiation cue or morphogen, all of those cells becoming another cell type A usually rarely happens when you're trying to differentiate stem cells in vitro. Instead, differentiation happens in a more probabilistic fashion where 30% of the cells will commit towards fate A, another 60% of the cells, for example, will commit to fate B, and another 10% might self-renew and do the same thing. And the way, or become the same cell type, and the way that cells can achieve this type of differentiation is actually through intrinsic fate bias. And so we think of the epigenetic landscape and replication as a potential source for this intrinsic variation. And this might play a really big role in maintaining the correct tissue compositions of cell types, where then self-assembly can take place to ultimately lead to the formation of complex organ systems. These things have been observed in other studies as how cells do this in development. So again, we're very interested in using nascent DNA methylation sequencing to establish a new epigenetic parameter. We're very interested in measuring this through different means, and I'll talk about some, and also modeling this, as I mentioned, with the Reed Lab. And also we're very interested in controlling it. So a lot of what's happened in terms of epigenetic editing has been looking at DNA methylation as a binary modification. It can be there or not there. But we're trying to develop biologics or molecules that can interfere with methylation, the reestablishment of methylation on a temporal scale by targeting this hemimethylated post-replication substrate. So we effectively are interested in using this tool as a first really demonstration in the context of cardiomyocyte differentiation. So again, this hemimethylated substrate being the target, and it's been known that small molecule inhibitors of methylation have been able to enhance cardiomyocyte differentiation in some contexts, but we know cells really can't withstand, especially somatic cells, can't withstand large losses in methylation because cells will ultimately apoptose if it does sort of lose methylation globally. And so what is the impact and what is actually changing in these small molecule inhibitors is clearly not a complete loss in methylation, unlikely. Could it be something on a more temporal scale in this post-replication space? And so to sort of get at this question, we've developed this deactivated DNMT1 molecule based on a point mutation known to make this enzyme catalytically inactive from prior literature effectively. And the unique thing about this point mutation is it actually still allows the protein to bind to hemimethylated DNA, but it doesn't have that catalytic activity. And so we expressed this in embryonic stem cells, and we've seen drops in global levels of methylation, which are indicative of slower sort of kinetics and remethylation. But we've also done cardiomyocyte differentiation preliminarily, And we see really strong enhancement in the activation of cardiac specific genes like cardiac troponin T compared to other DNA methylation inhibitors like RG108. And, in this case, we also look at the first day of beating, nothing too exciting to see here during this differentiation protocol in our particular system in hands. But at day 6 with this DDIMT1 express, you can see that we can actually get early and robust sort of contractility and beating within these cells. And so this is something we're really excited about to build on and understand actually the mechanism, if it is indeed through this sort of a prolonging of this post-replication remethylation of DNA and ultimately temporal regulatory variation. So the other side of this story that I want to talk about again is coming back. So this is largely motivated, and a lot of exciting work has started coming out about how DNA methylation really can allow us to form what are called epigenetic clocks, where we can, based on DNA methylation data from a blood sample, for example, we can predict one's chronological age pretty accurately. And so that's what this blue line is representing. But also we think methylation as maybe a marker of biological age, because individuals who seem to be above this line age more quickly and have diseases and incur diseases more quickly than people who are below this line, for example. And so this is the idea of chronological age being very different than one's biological age. And methylation seems to be able to be correlated with that, but the mechanism of really why and what's driving it is really poorly understood as well. And so, as I mentioned before, as DNMT1 can help to maintain this that a form of loss in methylation can occur through something called passive demethylation, where if you get rid of this methylation enzyme and cells replicate across another cycle before obviously this enzyme remethylates it, you'll create a fully unmethylated strand of DNA. And this is a substrate that DNMT1 actually has limited activity on. Most of its activity, again, is reserved for this hemimethylated state. And so you can lose methylation through 2 cycles of replication. And with that in mind, if you look at blood samples from newborn individuals, and this is a chromosome 8 and looking at methylation levels across that newborn's epigenome versus a centenarian, someone who lives to be 100 years or older, you tend to see this global hypomethylated landscape. And could this post-replication methylation delay actually be a source of where this sort of delay comes from is our main question in this regard. And so just to give you a flavor of what we see, when we look at our temporally dynamic tiles, again, the 10% being those that are less dynamic across replication, and the 90 to 100%, the Repli-DMRs being most dynamic, we can see this very nice positive correlation between how dynamic these regions of the genome are after replication and how susceptible they are to losses in methylation between those 2 individuals, a newborn and an aged individual, okay? And so it really suggests that regions that are more slowly remethylated after replication are also more susceptible to losses in methylation throughout the lifespan. So this is really sort of intriguing to us, especially as we think about regulatory mechanisms that might be aberrantly regulated during aging. And that's been shown in enhancer regions, for example, will lose methylation in some contexts as you go from young to old individuals, as well as the sort of global genomic landscape in general. So we are proposing this idea of epigenome replication as a 2-sided coin. Genome replication and epigenome replication on one hand promotes regulatory variation that can be really important for achieving things like probabilistic differentiation through molecular variation and heterogeneity. On the other hand, that replication can also leave our epigenome susceptible to epigenetic drift. And so we're interested in this substrate now as a target to both control cell fate decision makings and perhaps better understand aging, and also perhaps promote mechanisms of health span improvements and potentially longevity. So in summary, we posit that epigenome replication acts as an important source of temporal regulatory variation in cells, while simultaneously conferring susceptibility to epigenetic drift throughout the lifespan. So we're also doing some other things that I'm really excited about as well. We're trying to understand if this information of now understanding how methylation is reestablished after replication can actually help us to understand the activity and the behavior of how proteins and regulatory factors bind in this post-replication space. And we're trying to create something we call methylation pseudotime, and using long-read MinION nanopore sequencing to do this. And so the idea here being with Illumina, we get very short reads, and so we don't get a lot of CpGs per read. It's hard to really have a resolution of where that read might be in this post-replication trajectory. But by using longer reads, we can capture a lot more CpGs per read. And that will allow us to order these reads in a sort of pseudotime map. And so if we capture a read in space, we can say where it might fit within this pseudotime map. And if some proteins are more likely to engage with certain factors, we can sort of understand how those factors are actually binding. The other thing that we're interested in is actually being able to take snapshots of cell populations and understand how much delay they're experiencing, either through various environmental changes and metabolic shifts, for example.
Did we lose our speaker?
Yeah, I was about to say, Horace, I believe we may have had some network problems with Tim's connection.
Oh, goodness. Well, let's sit tight, see if he comes back. In the meantime, I can remind folks to ask questions in the Q&A. Feel free to upvote questions you see. I don't think you can downvote questions, but yeah, there's a good time to think about that. We'll see if he comes back. We've got a snowstorm over here. I was wondering if it was interfering with my end, but since you two are still on, I think it's not on my end.
All right, welcome back, Tim.
Thank you. Thank you. Sorry about that. I'm not sure what happened there, but I think we left off here. Is that right? Or let me share my screen, actually, so you can see where here is. Okay, can you see this?
Almost there. Now
Yep.
I can see it.
Okay. Where did I leave off? Were we talking about methylation pseudotime?
Yeah.
Yes.
Okay, great. And so I was just saying that we're encouraged because we can see this long tail that seems to appear when we look at the methylation level in long reads across a single read compared to what we would expect that read to be in bulk whole genome bisulfite sequencing data. So again, very hypomethylated in long reads. And one thing that we can also do is really arrest these cells again with nicotazole, and we can seemingly sort of minimize and help diminish that tail. So we're excited to look more into this. So for the last little bit, I do want to touch on some work that that we're doing in my lab that came from work that started in my PhD. So we're obviously very interested in how biomaterials help shape this paradigm of being able to control cell fate in different contexts where cells never exist in a vacuum. There's always some cell material interaction that is going to help shape our ability to be successful in regenerative medicine and controlling cell behavior. And so one of the sort of ways I really got interested in this sort of these questions actually is in the process of cell reprogramming. In my PhD with Song Lee, we demonstrated that there is this enhancement in reprogramming efficiency when we seed cells on topographies of elongated microgrooves, effectively from 40 microns down to 10 microns. And you can see that there's this increase as those grooves got more narrow. and these cells can express pluripotency genes. But in my lab now, we've been looking at a more sort of systematic way of understanding how these cell material interactions and even cell-cell interactions actually can help shape the reprogramming process. And so using this cell line that was developed by Davide Cacciarelli at Harvard University when I was there, they actually created an RNA-seq timeline of reprogramming of human fibroblasts across the reprogramming process. And so we took that RNA-seq and we asked, well, let's look at a set of genes known as the integrin and cadherin adhesomes. So these are all the genes that have been well characterized that help cells to bind and sense sort of interactions to their extracellular matrix and also other cells. And when we did that, we found about 103 dynamically expressed genes. And we developed an shRNA screen where we targeted 3 separate shRNAs for each gene. You can see that we get a lot of different profiles of changes in gene expression. Some genes and a large number of genes are rapidly downregulated very early in the reprogramming. Some are transiently upregulated, and actually SHRM3 is one that we're very interested in that seems to be transiently upregulated, while some seem to be more permanently upregulated. And so we performed this screen, and the first sort of thing that was very striking that we observed, here I'm showing a relative number of colonies, and that's relative to shRNA that are targeting genes that the cells don't express, so things like LAC-Z and GFP and RFP, for example. And what you can appreciate is that most of these knockdowns seem to be biased towards enhancing reprogramming, which we find really fascinating and was surprising at first. But we perhaps are thinking that maybe cells never evolved mechanosensitive sensing mechanisms that would actually promote this reprogramming process. But we've been looking more into this and actually looking at traction forces as well. And so we've mapped traction forces throughout this reprogramming process. And one of the surprising things and interesting things that we see, well, first we see that there are higher levels in early reprogramming that seem to dip. But what was very interesting to us is that we also see this increase in traction forces that seem to reemerge right around the time that, again, some of our top hits like SHRM3 seem to emerge as well in expression. And so we looked more into sort of how focal adhesions in particular in response to their binding to other extracellular matrix proteins. So we looked at uncoated dishes coated with things that are really can be beneficial for maintaining embryonic stem cells in feeder-free conditions like Geltrex, other substrates, sort of denatured collagen or gelatin, and fibronectin as well. And what you can see is that all of these have really kind of different, well, a group of these have different impacts on focal adhesion formation in terms of circularity, which is oftentimes associated, more elongation is associated with more mature focal adhesion formation. We also see a slight increase in area as well in these populations. And when we look at reprogramming, those same groups that are promoting very strong adhesions actually seem to be the most repressive to reprogramming. And we were kind of wondering, well, is this sort of a soluble thing? Are there just soluble ECM proteins that are around that are causing this? So we wanted to pattern ECM using some sort of phantom. And here you can see UCI, PDMS stamp sort of blocking the coating of Geltrex onto the surface. And then we remove the Geltrex and wash the surface. And then we seed the cells and start reprogramming. And you can see very clearly that the colonies are forming where that extracellular matrix actually hasn't been deposited, really suggesting that there's some functional impact on these adhesions. Just to on the reprogramming process. So we also looked at SHRM3, as I mentioned, one of our top hits, and SHRM3 really is showing us very interesting things as it relates to force generation compared to control cells, especially at day 12 when its expression peaks. But it also seems to be enriched for PI3K and MAPK signaling and perturbing that signaling, those signaling pathways. And that's really interesting because a lot of inhibitors of methylation, inhibitors that have been shown to enhance reprogramming have been PI3K and MAPK signaling related actually, but the implications of the impact of mechanobiology and mechanosensing in reprogramming actually has not been sort of attributed to why those things enhance the reprogramming process. And so just to wrap up this story, just wanted to touch on it, but we're very interested in understanding this rate limiting step of reprogramming. It's known that cells sort of go through this stochastic-like phase where only a small subset of cells transition to this fully reprogrammed state. And at the bottom corner here, I'm showing a GIF basically of how cells are interacting and engaging with their environment during reprogramming as a formation of a colony. And I think what you can appreciate is that these cells must be experiencing a multitude of forces as they are going through this reprogramming process. We're very interested in how the cell's ability to sense these mechanical cues actually contributes to their ability to efficiently do that. I don't have slides here, but I'll just say we're also very interested in how this might relate to cancer stemness and the acquisition of stemness more broadly. And so we've also been looking at work in that regard. So with that, I will end my presentation and certainly want to thank the people who have been doing the work. Julia Moraval is a senior PhD student who's been leading the work I talked about in the beginning, as well as with another member of my lab, Annie Tren, has been working to develop these epigenetic inhibitors, so to speak, that are targeting more of this temporal landscape, as well as Naveed Akhtar and Nandor Lasik, who are also a part of that earlier work that I mentioned. Andrew Fan has been leading that work on the reprogramming. And again, Elizabeth Reed and Honglei Ren, as well as Dr. Ching Ni, have been helping us and working with us on these, defining a kinetic rate parameter for every CPG in the genome, as I mentioned earlier. I'd like to thank my funding sources, and again, I'd be happy to take any questions. Thanks so much for the invitation.
Thanks, Tim. Yeah.
Thank you.
We had a bunch of questions. Maybe I'll start with the first one. I think one student was asking, what's the implication for this changes in methylation patterns for personalized medicine? How does it determine a patient's response to drugs? And I guess in general, can you talk about its impact on the age-related diseases? How you think it might impact those?
Yeah, great question. I think, well, we don't know yet, but what I speculate is that if it is a driver of sort of epigenetic drift, there are a lot of interesting studies, actually. One in particular that comes to mind, it actually looks at the rate of epigenetic drift across species in DNA methylation. And what you'll observe is that the rate of epigenetic drift as it relates to DNA methylation actually is linearly correlated with the lifespan of species. So if you look at mice, they have the fastest rate of epigenetic drift and they live the shortest. Monkeys are next in line, and then humans are very slow. And so we think it can be a great biomarker for perhaps predicting the progression of age and age-related diseases, but it also could be a potential target. And that's where it gets a little tricky because of the duality, again, of this post-replication delay that we're proposing. On one end, it's very important, seemingly, for perhaps regulatory variation and cells' ability to carry out their functions. At the same time, it leaves us vulnerable to this drift. And so can we target and limit the drift, and especially the drift that may be associated with faulty stem cell regulatory mechanisms or other impacts that age-related hypomethylated DNA and epigenetic features might have in promoting aging while preserving those other sides that are critical to stem cell function. So that's sort of our thoughts in terms of how we are thinking to approach this question from the clinical relevance of this post-replication delay.
So apparently there was another question about how do you maintain the same methylation pattern over subsequent transcriptions and minimize the variation?
Well, I think, one way to minimize the variation, if that's what you were wanting to do, in theory, could be to overload the cell with methyltransferase enzymes or epigenetic enzymes that can help maintain the epigenetic landscape. We're not sure if that's really what we want to do. We actually want to embrace the variability and see it really as a new, this as a new target in terms of controlling that variability and understanding how it allows for cell ratios to particular cell ratios that can emerge that ultimately will facilitate the formation of complex tissues. So we're more interested in embracing the variability rather than limiting it. But in theory, you can either get rid of the epigenetic modification completely, and if you don't have methylation somewhere, then you obviously can't have post-replication methylation kinetics there. There probably won't be much activity. But you can also keep it very high so that it's remethylated, perhaps by overexpressing enzymes, for example, responsible for that.
All right, great. We have a couple more questions. The next one I'll pose is that it's known that the composition of ECM can affect DNA methylation and potentiating differentiation. Is the conformation of the matrix or one of its constituents, is it the conformation that does this, or one of the constituents, and does it alter the degree of methylation of a specific gene or closely functionally associated genes?
I'm sorry, can you say that one more time? The beginning part I think I missed.
So it's known that the ECM composition can affect DNA methylation and potentiating differentiation. So is it the conformation of the matrix or one of the constituents that does it, or is it altering the degree of methylation of a specific gene or some closely associated gene?
Yeah, I mean, I think it's all of those things, really. So, in terms of how important cell adhesions are, certainly matrix conformations and compositions will play a big role in priming cells for certain differentiations. I think not a lot is known about how the epigenetic landscape is changing in response to those. So there's a lot of opportunity to really to better understand that. I think changes happen, semi-globally, but the locus-specific changes are probably the more impactful. So knowing exactly what is changing will allow you to infer some actual meaning to how a particular gene is going to be regulated effectively.
All right, we have time just for a couple more. Are there any tissue scale locus-specific epigenome editing systems you are interested in adopting or developing?
So we are doing mostly everything in vitro right now in a dish. But I think there are some cool systems, particularly in muscle, I think, by some folks at Berkeley that are trying to deliver, CRISPR gene editing to tissues. And the thing that I didn't really mention about this deactivated DNMT1 molecule that we're trying to develop is that we can tether it to a deactivated Cas9 and have local activity of this inhibitory activity. And that's something you could never do with a small molecule like 5-azacitidine or RG108, for example. So we're just trying to really see can CRISPR, deactivated CRISPR-Cas9 systems help us have this localized effect of this inhibitor as a next step.
Great, we'll move on to, in the sake of time. So the next question, does knockdown of the adhesome genes such as SHROOM3 impact the ability to accelerate entry and progression through the cell cycle?
That's a very interesting question. We haven't looked at cell cycle changes so much, but it would make some sense that could be the case, especially looking at the PI3K sort of enrichments that we've been seeing. And obviously cell cycle entry can have some role in the cell's ability to reprogram, but we haven't looked at that so much actually at this point.
Moving on, the next one is, does the higher rate of remethylation correlate to the expression of transition factors in charge of active transcription?
Let's see, correlate to the expression of transcription factors. So, we haven't looked at, the question was what? Can you say it again? I'm actually,
Sure.
okay, I see it here.
Yeah, if you want to.
In the data correlating remethylation rate to the expression of certain genes, does the higher rate of remethylation correlate to the expression of TFs in charge of active transcription, such as, so we did see very interesting enrichments in EP300, as you said. We're still looking through the data and trying to see, how does EP300 compare to that information we see in the, for example, the transcription data. But that's sort of the implication of it, right? Is that if there is an association with this EP300, perhaps there could be implications for at least the priming of substrates or gene regions for active transcription.
All right, so the last question that we might as well take since we're here, have you looked at histone modification and its temporal heterogeneity? In addition, on top of sort of the DNA methylation and temporal heterogeneity. So in terms of the epigenetics, looking at these other aspects.
Yeah, so we haven't done histone because we don't really like to do a chip with our BRDO IP, but we have been starting to look at accessibility in this context. And we do see some dynamics where we're not certain that methylation are necessarily driving those dynamics, but there does seem to indeed be some post-replication dynamics. And actually, other people have reported on that on the shorter time scales of our post-replication, sort of window. So there does seem to be changes, at least in the level of accessibility. But we are excited to look at histone modification changes too across cell cycle.
All right, so we can take the last one. How might a lack of abundance of methyl donors affect heterogeneity of methylation patterns?
Yeah, this is my favorite. I think that's what we wonder. And actually thinking about metabolic and Shreem, we were talking about the metabolic activity and SAM availability, which is a sort of a donor substrate for adding methyl groups. I think is going to have huge implications for the heterogeneity of those methylation patterns post-replication.
All right. Well, we are over time, so we'll end the question and answer period there. We can conclude just by simply thanking Tim for being available and sort of sharing your expertise with us. It's really been an exciting talk, and I greatly appreciate your making the time for us.
Thank you so much. Really enjoyed it. Really appreciate it. Yes.
All right. Have a great day, everyone.